Welcome everybody to JSEP 2020 presentations. It's good to see so many people, so thank you for joining us. I can see we have people from previous JSEP years and also families and friends, so great to see all of you. My name is Kasper and I take part in running the JSEP program on behalf of the government of Greenland. And a big thank you to the government of Greenland to prioritize education, also to support JSEP.
Yeah, welcome everyone. My name is Lauren Culler. I'm a faculty member at Dartmouth in the Environmental Studies program and at the Institute of Arctic Studies. I'm one of 4 principal investigators on the JSEP project, and it's really great to see so many familiar faces and friends here. I just want to upfront thank the Office of Polar Programs at the National Science Foundation for their support of JSEP through a grant to Dartmouth Institute of Arctic Studies.
And my name is Erica Wallstrom. I'm a high school educator and on the Dartmouth team as well in the United States. And we work collaboratively throughout the year in order to build strong connections with these real-world polar scientists and instituting the best practices in secondary education. So that's the model for JSEP that we strive for.
Yeah, and as we all know, 2020 has been a challenging year in many ways, and it's a lot of young people have missed out on that behalf. So we are especially happy this year to be able to bring 50 young people together to do science and to meet up across nations. So both here in Kangerlussuaq physically, and also the students participating virtually from the US.
It was really challenging to transition with on the go as the travel restrictions were put in place. And this transition was only possible because of such a really strong and collaborative team in order to put together the most dynamic learning experience, even though many of us were not able to travel there physically. So we just want to give some shout outs to the amazing people that are on our team. First of all of our amazing students that have remained engaged and positive and ready to learn and to share despite the challenges and maybe disappointments of not being able to travel to Greenland physically. We have a student teacher who's participating from Greenland, and so really thinking about building capacity for inquiry-based learning in all of our classrooms across our different countries. Our graduate fellows have put in timeless hours trying to put together the most dynamic lessons and facilitating the research project. We have an amazing team of educators and mentors that are from all 3 countries and span through high school or the gymnasiums all the way through the universities. And lastly, I just want to really thank all of the friends and family that are joining us today and supporting our students from home and from afar, and making this all possible for them to be here today.
Yeah, also thank you to field support we've been having here in Kanger. We've been in quarantine, so no visits from outside, but we have had Chris to help us with the logistics, and also Nini to help us with the food. It has been impossible to do JSEP without these guys. And also big thank you to Natalie for putting together all the technical stuff. It's a big challenge this year. And a thank you also to CPS for supporting us on logistics, all the gear we have here at JSEP. So thanks, everybody.
So just to give a little bit of context for all of the guests. So last week, the team of 50 students came together and students completed interactive polar science modules led by Dartmouth graduate students. And this week, since only Monday, they've been working on collaborative field research projects. So in just 3 days, they've gone from start to finish, designed and completed a project. And today is the culmination of that experience through the presentations you're about to see. So we're really excited to see and share their great work. Just briefly, so when students are presenting, we kindly ask everyone to keep their microphone muted. And we are going to use the chat feature of Zoom for questions. If you can, hold your questions until the presentation is done, and then you can type it into the chat. and I'll be able to read those and hopefully ask 1 or 2 questions to each group. We'll also briefly unmute everybody at the end of each presentation so we can give a virtual round of applause to everyone. So without further ado, we're going to jump into the first group to present. This is a group with students in Greenland, from Denmark and Greenland, and students from the US working on a project about ice and engineering. And I'll turn it over to the ice group.
Yeah, welcome to the ice group.
Hi, so let's just jump right into the ICE project. Next slide, please. So, preserving glaciers and keeping the planet cool. Glaciers are actually a major climate change, or a keeper of climate, so to say, as in they have a very, very, very bright surface, which reflects a lot of light, and they are very, very cold, both of which are factors to keep the environment in which they are placed very, very cold. But as of lately, both with the trend of global warming and also other local climate changes, there's begun to be in this positive feedback loop between a lot of glaciers, namely in the fact that they are very, very bright, and as they melt, a lot of water or dark area around them is exposed, which leads to more heat absorption from the sunlight, which heats up the environment, etc., etc. Next slide.
Yes. So our place for field work at point 660 is some distance from Kangerlussuaq, our town. The trip from the town to ICET is 35 kilometers, and it takes about one and a half hour to go there. And we are 11 students in all, yeah, in 2 groups and doing the research. Next slide. Yes.
So within this project, we were all concerned with the same idea. So understanding the factors that could lead to increased melt rate and kind of like coming up with ideas for how we might combat that effect and preserve glacial ice. So to pursue this one idea, we split into 3 separate groups to take different approaches. So the field group, which was in Kanger, was studying how the thickness of sediments on cames could affect the melt rate of the ice underneath. Then there was the remote lab group, which used ice cubes and sediments and soil to kind of model how the different factors of sediment cover could affect melt rate. And then you had the third team, which was also in Kanger, and then also had a remote student, the engineering team. And they kind of built on the work of the previous 2 groups to kind of study how we might be able to come up with practical real world solutions for slowing down the melting of glaciers. Next slide.
All right, the lab team. So the lab team looked at how the sediment on top of the ice affects the ice's melting rate. We chose 3 characteristics that could be tested with the resources available to us. Specifically, we looked at the thickness of the layer of sediment, the size of the particles that make up the sediment, and the proportion of the surface of the ice that is covered by the sediment. Conducting these tests allowed us to isolate the variables in play on the glaciers and determine what role each one has in contributing to the total melting of the ice. Next slide, please. Here you can see our 3 setups spread across the country. Next slide. All right, so in this experiment, I looked at what influence the size of the sediment has on the ice's melting rate. I concluded that the smaller the particles in the sediment, the faster the ice melted. This, I hypothesize, is due to the density of the sediment, with smaller particles packing closer together. This reduces the amount of insulating air within the sediment and allows the heat to transfer faster to the ice. As you can see in this graph, with the yellow line being the small sediments, it melted the quickest of all of them, with big sediments coming in second. In the case of the fine sediment or soil, there is only a slight decrease in melt rate from the control with no coverage. And this could be due to the amount of air trapped in the soft, fine soil. Now, ideally, in the future with these experiments, they would be done with sediment of the same composition. the only difference being the size of the particles in each, as opposed to having 2 with rocks, 1 with soil. And it would also be done with more trials and more steps between the sediment sizes to get a better picture of the whole scene. Next slide, please.
For my part of the lab experiment, I analyzed how the percentage of ice covered impacts the melting rate. I conducted 5 different trials, each adding on another quarter of coverage per trial. I used black paper to make each quarter very defined. The black paper simulates the effect sediment would have. As you can see from the graph, the blue line representing no coverage has a lower melting point than the orange full coverage line. Another interesting thing to see was that every time another quarter of coverage was added, the melting rate increased. This was an interesting relationship I saw in the data. In all, the overall results point to a higher percentage of coverage does in fact increase the melting rate.
So in my experimental trials, I was looking at how increasing the depth of the soil layer would change the melt rate of the ice beneath. So as you can see from my data, the 2 tablespoons of soil, which was the thickest soil, was definitely the one that had the fastest melt rate in the beginning. And then the one with 1 tablespoon and the control were both sort of in the middle, and then they all kind of leveled off at the end. So in theory, a good explanation for this would just be the albedo effect. So if you have this super reflective surface, which is just the control, which is just ice, and then you add on layers of soil, which are darker, those are going to absorb more heat, and so the ice will melt faster. And that definitely makes sense. But we also kind of think that since the 2 tablespoons, so the thickest layer, was melting at a rate that was so much greater than the 1 tablespoon, we also kind of think there was another factor involved, which was the conduction just from the soil itself. So I had left the soil and the sediments in a pot in the shade, so they weren't excessively hot. But the thing is that they were still, quite a bit warmer than the ice that had come right out of the freezer. So we think that whatever effects we would have seen from the albedo just having a darker surface was also kind of conflated with the extra variable of just having the conduction from the soil that was just warmer than the ice. So if we were able to kind of continue this and do this on a broader scale with more trials and different varying thicknesses, what we hypothesized we would eventually see is that we would reach a point where you would kind of get an insulation effect coming into play, where you would kind of have this increasing melt rate as you increase thickness because of the albedo, but then you'd reach a point where it's sort of insulating the ice, and so you'd kind of reach a peak and then go back down as that comes into effect. So if we were able to do more trials and kind of have a bigger setup, that's something that we would definitely have liked to explore and kind of get a better idea of the interplay between albedo and insulation. And those are both ideas that you're going to be hearing a lot about when we go to our 2 teams in Greenland. So speaking of them, can we go to the next slide? So the lab team discovered that the sediment thickness, size, and percentage of coverage all did affect the melt rate. So how does that translate to our groups in Greenland? Next slide, please.
Okay, so in our project, we wondered or researched the difference in sediment thickness of the cames at 0.660. And in the video you are seeing, we are standing on the edge of the ice at 0.660, you can see the cames, which Erica is pointing at, and they are these cone-shaped brown things, which is created by the melt from the ice sheet, they are covered by sediment or dirt, and this sediment has an insulating effect, and therefore the ice under is not melted, and this is what we are interested in. So next slide. And therefore our question was, what role does the sediment play in the creation or preservation of the cane?
So How did we do it? We measured height of the dirt sediment, thickness of dirt sediment, orientation north, south, east, and west, size of the cames, experiment before and after pictures. Next.
Next slide. Yeah, analysis and findings. This is the picture of the where we are working in the field. As you can see, the round ones are the cames. Yeah, next.
So when we were near the cames, we measured, we took samples from them. We took sediments from each of the 4 orientations, and it was a 10 by 10 centimeter, so we could compare the data. And then we summed it all up, and this is a diagram showing the results. So as you can see, the southern facing side has twice as much sediment placed on it as the northern side, which is quite interesting. Next slide, please. Also, we can see that the thickness of the sediment is actually greatest on the southern side of the came. Next slide, please. And this can be explained maybe by our qualitative experiment where we took before and after pictures of this came. So if you could press, Erica, yeah. So on the came, we removed a lot of the sediment on some of the came. And then when we came back 24 hours later, a lot of it was melted away. And then we could see that all of the sediment that had been placed on the northern side actually moved to the southern side. So yeah, next slide.
Yeah, we also found that the average thickness of the sediment was between 1 to 2 centimeters regardless of the size of the cames. And so we believe that this is the sweet spot where the sediment actually becomes insulating and starts protecting the ice and helps it not melt. Next.
So the conclusion, and sediment reduces melt rate. The sediment is cames tended to have a thickness between 1 to 2 centimeters. We believe sediment is needed to preserve the ice underneath. We also found that the southern face of the came had a tendency to have a thicker sediment. To conclude exactly how sediment can affect the preservation of cames, further research is needed. Next slide. So I've been drawing and painting JSEP2020 ice field team. Next slide.
So now the engineers come in, going on to protect the glaciers. So how can we preserve these melting glaciers? Is there anything we can do to prevent them melting? Next slide, please. So as engineers, what we do is we look at different properties of materials to determine what would be best. In this case, we had a couple of hypotheses. Hypothesis number 1 would be that materials with high albedo, at least theoretically, should reflect a whole lot more light and therefore keep the heating from the sun from becoming a problem on the glacier itself. Another one was that materials from nature could be the solution, since nature usually finds a solution before we do, and it's usually a lot more efficient. Another last thing we decided to try was to see if we could find something cheap and accessible. So we also brought a simple sheet of plastic out on the ice. Don't worry, we retrieved it to see what difference that would do. Next slide, please.
Yep, so we got out there and set up our project. We prepared 5 different materials to work as covers. You can see them on the pictures to the right. We got a plastic bag, black plastic bag, sealskin, just a simple white towel, aluminum foil, and about 3 centimeter layer of gravel, which we also found on the ice. Then we placed our covers on the ice and came back the next day, we measured how much the ice has melted underneath our covers, and then we also measured the albedo of our covers. Next slide. From the results, we can say that aluminium provided the best protection because, as you can see on the graph, no snow has melted underneath the aluminum. We can explain that by two things. First of all, aluminium had one of the highest albedo factors in all of our 5 covers, and second of all, we accidentally trapped an air bubble in between the 2 layers of aluminium foil, which provided better isolation. If you keep looking at the graph, you'll see how much ice has melted onto the different covers. There's names underneath, and the red line will be symbolizing how much ice without covers melts on average, which is about 4.4 centimeters. While measuring our albedo, we stumble upon a funny thing, which is that there is a very thin connection between the albedo of the material and how much it protects the ice, which means if a certain material, for example, a towel, has a very high albedo or reflective capability, it will not necessarily mean that it will preserve the ice best. A plastic bag was a very good example of that, as it had the lowest albedo, yet did one of the best jobs. Next slide.
So what we can basically conclude from this is that both air, or insulation factor, as we call it, and the albedo has a major role to play in reducing ice melt. There was no direct correlation from our data that actually suggested that albedo was a main factor, but what we definitely could do was to work further towards researching the difference in albedo with more non-isolating factors and just different albedos, or we could work more with the insulating properties of different materials by taking different materials with the same albedo, but with different amount of insulation. Next slide, please. This was a lovely experiment done by Wallace. So I would like to thank her for that, but I'm afraid I can't explain a lot of this. So next slide, please. So essentially what we've drawn from all of these 3 experiments is, number 1, preserving glaciers is really important. They have a major role to play both in the global and local climates. And what we can take away from these different experiments is we know a lot more about the properties of ice and sediment on top and general melt rates. And we know that it's possible to cover these glaciers and reduce ice melt from different projects that have been done in the Alps, namely in Austria and Italy. However, those were only to partially prevent melt. And also beyond our great research that we've done, we also found out that we need a whole lot more before we can draw any serious conclusions and come up with some evidence that would be good enough to at least legitimize ourselves. But that is all. Thank you.
Thank you so much. So maybe Natalie can unmute everyone briefly so we can give a round of applause, or you can unmute yourselves.
Awesome.
Excellent, thank you so much. And I think that we have maybe about time for like 1 question. So if anybody has a question for the ICE group, they can put it into the chat. And in the meantime, Erica is going to queue up the next presentation, which is going to be by the Food Sustainability Group. There's a question from Maddie in the chat. I want to know, how did you measure how much ice melted under the objects you put on the ice?
I think this is for our group, right? For the lab group.
It was actually for the, well, I guess you guys could answer it too, but I was curious also about the
Oh, for the field group.
The field group who was measuring things on the glacier.
Right. Yeah, why don't I let them take that one, because I think that's the more interesting answer.
So essentially what we did was that we drilled a hole, measured the depth before and after we had placed it. So we played, oh, that sounds dumb, the day before and the day after. We drilled a hole underneath the cover, put the cover on, drilled a hole, measured the depth, put the cover on, left for a day, came back, took the cover off, and measured the depth. We did, however, also, just to make sure, drilled holes besides each one, so we had a reference point, which is the one we've drawn the average from on the graph. But yeah, that's how we measured it. Of course, there will be uncertainties in that, but that was the most reliable way we could think of.
Great, thank you so much. So thank you so much again to the ICE team. Really cool presentation and great start to today. We're going to move on. We have another group that's been studying food and livelihoods with Shauna Bulger from Dartmouth, and they have two shorter presentations. The first one is what's on your plate, and I'll turn it over to them.
All right. Hey guys, welcome to our presentation. My name is Victoria Larcon Macias, and I am from Santa Fe, New Mexico.
Hi everyone, my name is Kikai Del Mendo and I'm from Hawaii.
Hi, my name is Kira McLeod and I'm from Plano, Texas.
Hi, my name is Miriam Scott and I'm from Eagle River, Alaska.
And I'm Mani from Denver, Colorado.
Okay, thank you. Our group was a part of the food module with Shauna and Angie, and the title of our project is What's on Your Plate?
Yeah, so for some background on our research, we wanted to study the relationship between traditions, culture, food, and the carbon footprints that comes from commercial airlines, shipments, and other modes of transportation. We wanted to preserve tradition while reducing the negative environmental impacts that come from these commercial transcontinental trades. Next slide, please. So the question that we wanted to ask was, what staple foods are coming from other areas, and are there more traditional and or sustainable foods available? Next slide, please.
So some of the methods we used was call our local grocery stores and see their most popular items, such as fruit and vegetable produce and non-perishable food items, as well as animal products. So towards the end, we gathered the data and analyzed our most popular items in our regions by using word clouds and calculating the food's miles. Next slide.
So we reached out to 27 grocers and only 10 responded to the survey. The most purchased items from this larger infographic in the center represents the most purchased items from all tested geographic regions. And then the most mentioned popular food items were bananas, tomatoes, and potatoes. Next slide. And we further broke down the larger infographic into smaller geographic regions so we could calculate the food miles. And food miles are the miles that a food item is transported during the journey from producer to consumer to show CO2 emissions that contribute to climate change. And then we calculated the top staple food items from each region. So for Eagle River, Alaska, the top food mile total was 9,000. For Colorado, the top was 3,000. Next slide, please. And then for New Mexico, the total was 6,000. And for Hawaii, the total was 300. Next slide, please. And then for Texas, it was 4,000 miles. And our key findings were that bananas are the most imported produce item nationwide. We also learned that Hawaii's produce was mostly local, which makes sense because of the tropical environment and longer growing season. Other regions that we tested imported their popular foods, and there were not a lot of sustainable options nationwide because of the long distances they had to travel to get to the grocery store. Next slide.
Hi, everyone. Again, my name is Kikai, and I'm going to be talking a little about sustainable options with respect to our food and where it comes from. The most important idea is to buy local. Buying local is the most effective way to decrease your food miles or decrease the distance that your food takes to get to your plate. And buying from local businesses reduces the carbon footprint of your food, and it also supports local farmers. Next slide, please.
Hi, again, my name is Kira McLeod, and I'll be talking about the significance of getting locally sourced food. So first off, it helps the environment. So the distance that it takes imported food or food that is not grown locally to get to the grocery store has significantly more food miles than locally sourced food, which increases CO2 emissions and increases the carbon footprint and greenhouse gases. So by getting locally sourced food, we can cut down on that. Also, locally sourced farms tend to use less pesticides depending on the crop. It also stimulates the local economy by keeping the money in the local economy rather than moving that money to another country for imported food. And it also cuts out that middleman and makes sure that our farmers are getting the money to sustain the business. It also creates jobs, which also stimulates the economy. Next slide, please. So for the conclusion, we figured out that our most top produce from our general areas were imported from out of the country. So our top, especially bananas, which were from Ecuador, and which I said earlier, that increases the emissions when compared to the emissions to the local travel. And for our responses, we figured out that Hawaii and Alaska have more local food chains, grocery stores than Texas, New Mexico, and Colorado, which we think had something to do with getting our results. When it came to Hawaii and Alaska, they were more successful, which I think had to mostly do with the fact that they had more tighter knit communities rather than Texas, New Mexico, and Colorado, which tend to be more urban areas. And with regional chains, they weren't in national chains. They weren't as interested in figuring out where the food came from or having a relationship with the local farmers. And so we think something we do differently next time is give the survey online for stores to fill out instead of calling them, because it seems kind of solicity and it was a little bit awkward to get a hold of them because of the times that we called them at. So we would do that. Next slide, please.
Thank you, everyone who helped to make this amazing program possible. We want to thank the Greenland, Denmark, and USA teams and educators for creating a diverse learning environment, and as well as a huge thank you to Shauna and Angie who helped lead our group. And we hope you learned more about the importance of food sustainability.
Thank you so much. So maybe we can do a very quick round of applause before moving right on to the next food presentation. We'll hold questions for this group until the second food group is gone, and then if there's time, we'll have questions then.
Yes, we are Emily, Inoko, Julia, and Omar, and our title is How to Value Surrounding Food Chains Between Generations. Next slide, please. We wanted to know what influences people's food choices, and if it differed between the age group. Some of the potential influences we considered are environmental impact, cultural meaning, and health benefits. Next slide, please.
So the first factor we looked at was cultural impact, and the ability of, maybe traditional or family recipes, as well as just culture in a broader sense and regionality to determine what people eat. Next slide.
The second impact we looked at was health, and we thought it was going to dramatically change your diet because it helps you reach or maintain healthy weights, reduces your risk for chronic diseases, and promotes your overall health. So we thought this would be pretty different between the age groups, but we were surprised. Next slide.
We also looked at environmental impact, which the previous group focused quite a bit on. So kind of a fun fact is that livestock and their byproducts account for 51% of all worldwide greenhouse gas emissions. So that's specific to, of course, animal byproducts and animal products. But as we learned from the previous group, local produce also has really good sustainability measures compared to imported. Oh, next slide. So the method we used was the Q method, and you can go to the next slide if you'd like. So in the Q method, you have participants rank their agreement or disagreement with a statement. So we averaged out people's agreement and disagreement using a point system. So if the mean was positive, that means they in general agreed with the statement. If the mean was negative, that means they in general disagreed. And of course, the closest to 0, the less opinionated and the more neutral they were. Next slide.
So the data for culture, the statement that we put was, I choose my food based on its cultural values, family, and or traditional meaning to me. And for the people under 18, around 88% of them were either neutral or disagreed that their diets were influenced by their culture, while 80% of the 60 plus people agreed or strongly agreed that they chose foods based on their cultural values. And as you see on the top, the mean was negative 0.18 and the over 60 mean was 0.90. Slide. Next slide.
So for the next Q statement, we said, I choose my food based on its health benefits and or because of my dietary restrictions. So this included health benefits that you just wanted to be healthier, or if you had gluten allergies or some kind of health condition. So under 18, the mean was 0.0, and for over 60, the mean was 0.7. And this was really surprising to us because they were so close compared to the other ones. Next slide.
Environmental impact had the largest discrepancy in means. So the statement was, I choose my food based on its environmental impact and or contribution to climate change. The under 18 group generally agreed with a mean of 0.75, and the over 60 group generally disagreed with this statement with a mean of negative 0.4. Next slide, sorry.
So here are our findings. For the cultural side, as we said earlier, 80% of people agreed that culture did affect their diets if they were on the older side of the generation, while younger people slightly disagreed that culture impacted their food choices. Based on health, it was pretty much an even split. Both young and older people agreed that health impacts your food choice greatly. The younger people agreed slightly more. And environment was the biggest gap, with the difference between 1.15, the largest. And it basically means that younger people agreed that the consequences of global warming and the environment determines what they eat, where older people disagreed that their food choices were based on environmental impact. Next.
So as soon as we finished this experiment, we took a step back and we tried to see what potential factors influence the data we got. And We each called or surveyed people from around the country with the exception of the northern region and the central region. And we also looked to people in Greenland with the exception of the northern region and the eastern region. So we had a good amount of people that we surveyed. What we found though was that we had a big discrepancy between rural surveyors and urban surveyors. 40% of older people lived in rural areas, whereas only 12% of the people we surveyed who were younger lived in the rural areas. There were generational differences because culture values may decrease over generations and awareness of climate change has increased in the younger generation. So when we were looking at the geographical differences in our data on an individual basis, we found that those in the eastern region were more likely to agree that climate was the most important factor when they went to the grocery store. For the southern part of the US, it was culture, and for the western region of the United States, it was health. In Greenland, we found that culture and climate were split in the western region, and health was the most prevalent factor in their diets in the south. Next slide. So thank you so much to Angie and Shauna and all the mentors in Kanger and in the US for all their support, and we learned a lot, so thank you.
All right, time for another round of applause.
Yay.
Thank you so much. So we have a few minutes, so if people have questions, they can put those questions into the chat and I'll read them to the group. And in the meantime, I think Erica will start to get the next presentation about biodiversity queued up. So feel free to send your questions. There's a question from Celia to the food groups. How did you survey people and who did you survey?
We surveyed people through Google Forms. So we created like 5 questions. And then before those 5 questions, we asked what their age was and where they lived geographically. And then after that, we were able to just take the responses from that and put them into graphs and such. And we only asked exclusively under 18 and over 60.
Have another couple minutes and I will just say that there's lots of great feedback coming in about all of these presentations so far. So great job, everyone. Any other last questions for the food groups? Clara has a question and she would like to know from the first group who presented, how would you relate what you learned to the Arctic?
So I'll answer this because this was sort of a question I was thinking about because I live in Alaska and that is in the Arctic. And I have family and friends who live in northern Alaska and it's an issue to get food up there because of how far it is. And there's nothing that grows in like Uktiavik, which is complete ice. So what I learned from this is it's best for like the Arctic to either transport food from their region that's local, but there isn't a whole lot that we can do. Maybe in my region, we have greenhouses, which are sustainable options, but that's sort of like it.
Thank you, Miriam, for answering that. And there are more questions coming in too. We're gonna have to move on. And I just wanna also really briefly say it's very wonderful to see so many JSEP alumni who are saying hello in the chat. So welcome to all of our JSEP alumni. It's great to have you back. Yeah, thanks for being here and sharing this experience. So we're gonna move on to the next group. A group of students from the US and 3 students who were based in Greenland, 2 from Greenland and 1 from Denmark, working in the field. And this group, the fish group, has been working on aquatic biodiversity. So I'll turn it over to them.
Oh, sorry. Welcome to our presentation made by the first group. Next, please. What is the point of the whole result? Well, the point is to find out the effect of temperature and precipitation on biodiversity, and we intend to answer our central question: how do the differences temperature and precipitation affect the biodiversity in different aquatic habitats with the information we collected? Next.
So why we chose this question, there were a lot of considerations that we looked at when we were constructing our question. We really wanted to take advantage of kind of how spread out we all were geographically. We're located all over the US and then also Greenland. So we're across 6 different biomes collectively. We also, thinking about climate change, wanted to kind of look into how future environmental changes could affect biodiversity in different biomes and the places where we live. And then finally, we chose to study aquatic macroinvertebrae because they're good bioindicators, meaning that they're very sensitive to environmental changes like temperature and precipitation, which is what we studied. And so they were really good organisms for us to work with. Next. And we wanted to go over some important vocabulary that'll be popping up throughout the presentation. So first, biodiversity is the variety of life in a particular habitat or ecosystem. A biome is a large region of Earth that has a certain climate, and temperature and precipitation are kind of key characteristics in determining a biome. Macroinvertebrae are organisms without backbones, and they're visible without the aid of a microscope. And we specifically looked at aquatic macroinvertebrae, which just live near rocks and sediment on the bottom of lakes, rivers, and streams. Precipitation is rain, snow, sleet, or hail that falls to the ground. And finally, fish stocking is when fish are released into a river, lake, or ocean to either increase existing populations or create a population where there wasn't one before.
All right, so our methods. Well, first of all, what materials and data did we use? Well, since we're not all here up in Greenland, we didn't have the opportunity to use the exact same tools. So just for a comparison, we have 2 different cups in the right top and right bottom. So as you can guess, our results varied a bit. But we also had to take our samples, we had to get some containers, in our case plastic bags or other stuff we could contain our samples in. But up here in Kangerlussuaq, Greenland, and Penny, I know, she used a thermometer and microscope as well for this research. And how did we do our sampling? Erica, can I get you to press twice? Thank you. Welcome to How to Do Fieldwork 101. Well, first of all, we started by disturbing the water for 15 seconds, and then we scooped 5 cups of water and dirt. Whatever landed in the cup, we took, stuffed it in the container, and then we moved on and went home, where we would put everything out on the table in a cup or something, and just find everything that was alive, and then we would identify it. As well as all this physical equipment, we also had something called neon data, which we will cover later. But just to get something of it out now, it's something that provides us with more data, which will lead to better and more precise results. So how does this help our research? Well, our fieldwork, normally the fieldwork is used for better understanding how natural environments and it's used for better understanding how the natural environment and its functions. And in our case, we needed to understand the macroorganisms we were working with. And by doing so, we got an understanding of what made their ponds or lakes
so habitable. And by doing this, we covered a bit of our conclusion. Next slide, please.
So NEON data is data from the National Ecological Observatory Network, where we saw the organisms in 7 lakes across the United States, from Alaska to Florida, and the microorganisms that were in those lakes. With this information, we could see the biodiversity of the lakes and then compare them to the ones that we got in our field data with the people that went to the different lakes around their country. So the way that we use this data, it was mainly as a way of creating a similar data set to the one that we were getting. So that way we could compare results, and that would help us reach a better, more structured conclusion. Next.
So if you look at the graph on the left, it shows our group's biome placement relative to temperature and precipitation. And as you can see, the tropical seasonal forest and savanna has the most precipitation and hottest temperature, while the tundra has the least precipitation and the lowest temperature. The woodlands, shrublands, and temperate seasonal forests are closer to the middle. We were able to test a very diverse biome range, as our group is very spread out over the US and Greenland, which was good. Next slide. Here is a graph showing the correlation between the average temperature at each individual site and the number of orders we found. And when we say orders, we mean the specific type of species classified. And this data suggests that as temperature increases, so does the biodiversity. Next slide. Similar to the previous graph, here we have one comparing annual precipitation from each biome location tested to the number of orders found at the sites. Again, this data suggests that as precipitation increases, so does the biodiversity. Next. And here we have a bar graph of the number of orders we found in our fieldwork relative to our biome type. The hot and wet tropical seasonal forest and savanna contain the most number of orders, while the cold and dry tundra contain the least number of orders. It is clear, looking at this graph, that as the biome temperature and precipitation increased, the number of orders increased, and vice versa. Next.
So the results that we were able to draw from both our field data as well as the data that we collected through the NEON database is, number one, that the NEON data did not show any discernible trend relative to temperature and precipitation, neither positive or negative compared to biodiversity. Our field data, nevertheless, showed a positive correlation by both temperature and precipitation to regards to biodiversity. And third, we were also able to see that colder biomes tend to have less biodiversity than hotter biomes, which correlates to what the data we collected. Next.
Hi, so here are some examples of samples taken by JSEP students, and the two, like towards the left, were from the group in Greenland. They found a ram snail and an aquatic worm, as well as a case-building caddisfly larva, which was a pretty common thing to find, it seems like. And someone also found some mayfly nymphs. Next.
Hi. So the significance of our results is that overall our findings suggested that there was a general trend which showed a link between precipitation and temperature increase and greater biodiversity. This trend is common across the globe and can be seen due to the fact that there is an increase of biodiversity shown moving from the colder polar regions to the humid tropic zones of the equatorial region. Other studies hypothesize that the trend could be linked to primary productivity, which is the rate that photosynthetic organisms can produce organic compounds from CO2 in an ecosystem. And other studies tested the relationship with habitat size or temperature, like us. Next slide.
So the other part of our scientific question had to do with how biodiversity relates to climate change. So how can this information be interpreted with respect to the future and climate change? So we know that as environmental conditions shift, species will be forced to adapt or migrate. So even seemingly minor changes in rainfall patterns, precipitation, or temperature fluctuations of just a few degrees could have major impacts on an ecosystem. So as illustrated by the diagrams towards the right, ecosystems are deeply interconnected. So even the loss of a single species, particularly if it's a so-called keystone species, could have devastating impacts on biodiversity. Next, sorry.
So these two charts show the impact of climate change across the globe. As you can see on the left, changes in precipitation will occur, most significantly the increase of rainfall in the winter and corresponding decrease in the summer. On the right, you can see the temperature changes that occur, which overall depicts a temperature increase. Next.
Okay, so how does the temperature change affect aquatic insects and fish? So aquatic insects' ideal temperature to function are from 7.2 degrees Celsius to 18.3 degrees Celsius. And a temperature increase for fish means reduction in the number of wild fish and introducing more predators for them. So the increase of temperature is forcing insects to change their behavior to survive. And that affects the fish because the fish mainly prey on these aquatic insects. So it's causing this whole chain reaction. Next slide. So the overall effect of climate change on ecosystems. Climate change is causing alterations in average temperature and rainfall yearly. This forces organisms to either adapt, migrate, or die. Adaptation to changing climate by species like aquatic insects causes a chain reaction in the ecosystem. Eventually, all animals who rely on aquatic insects as food source need to adapt. The future of global biodiversity is dependent on species' ability to adapt. If species can adapt, then biodiversity will be preserved. If not, biodiversity will suffer. Next slide.
Sources of error. Pollution, human activity, for example, trash. Proximity to town cities, like if you are closer to cities and towns, there's a high chance that there will be more trash and more pollution. And fish stocking and other factors not measured, and data uncertainty. For example, human error and not similar equipment. Lack of variety in independent variables. For example, the neon data covered less different kind of biomes compared to the fish group. Yeah, next.
All in all, our research led for many questions to arise, such as whether human intervention has a notable effect on biodiversity, such as pollution and stocking, but will this ever reach a magnitude so that it eclipses the effect of temperature and precipitation? Also, are there more unrecognized or unresearched factors that contribute to the amount of biodiversity in a certain habitat? Finally, will humans in the future be able to utilize science and technology to engineer solutions which allow us to combat the negative effects of climate change on biodiversity? That's the end of our presentation. Thank you so much to Maddie and Lauren and Reggae for their guidance, and to all the mentors for making this all possible.
Thank you so much. So we can unmute for a round of applause.
Whoa, 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 whoa.
And if there are questions, please put those into the chat. There's already one coming in. The first question from Robert Baker is, will climate warming trends lead to more biodiversity?
Well, I can take this one. So as the temperature and precipitation change, I mean, it's going to force species to adapt. So if the species can adapt, then they'll survive. But some species might not be able to adapt, and they might end up dying out and going extinct. So we don't really know at this point, but most likely it will not increase biodiversity.
For the answer bar, other questions for the aquatic group? Celia asks, why is it harder for fish to live with just a few degrees increase in temperature?
All right, I can actually answer this one. Well, if you have microorganisms, you need something to eat them, but they are very fragile, which means the hotter it gets, they need to adapt faster. And if they can't, they will die out, and the fish's food sources will vanish as well. And therefore, just a few degrees in temperature change can lead to extinction.
Thank you, Julius. Any other questions for the aquatics group? So here, one just came in. Do we have any examples of present day fish extinction? Does anyone know? Anyone want to take that one? Maybe you can think about it. We're actually headed into a break right now. So maybe we can unmute and just another round of applause for all of the presentations so far.
Yay.
So we have just under 15 minutes. So presentations are going to resume at 1:15. And we recommend anyone who's planning to stay on for the presentations to just stay in the Zoom room and just maybe turn off your camera, make sure you're muted, and feel free to take a break and then make sure you're back at 1:15. And we have 2 more presentations about soils, and then the final presentation is about the ecology of sound. So we'll see you in about 15 minutes. Thank you all for being here. Okay, everyone, just so you know, presentations will resume in just about a minute. Okay, welcome back, everyone. If you're rejoining us, we're gonna move into the next presentation. There's a group of students from the US, mentored by Amelia Fitch and Matt Ayers from Dartmouth, who've been working on understanding soils and life beneath our feet. So I'd like to turn it over to the soils team to get going with their presentation.
Hi everyone, and welcome to our presentation about soils, which we have titled Sedimental Connections, as a little play on words there on sentimental, because we all have soil and we're all connected by earth and plant life materials. So we dug deeper and understood and compared the particulates that bear life, and the students in this group were Katie, Alicia, Chloe, Roman, Celia, Anna, Samuel, and Quinn, and then our lovely mentors were Amelia and Matt. Next slide, please.
So before we dig into our project, we wanted to share a quote that we think really captures the importance that soil has both indirectly and directly on all our lives. Essentially, all life depends upon soil. There can be no life without soil and no soil without life. Next slide, please. Just a little about our project. So human impacted and natural soil from across different regions in the US was examined and analyzed. So in each of these unique regions, we examined and analyzed two soil sites, I guess. One from a human impacted area. So that would be an area like a lawn at your home, or just anywhere where humans have altered the environment. And the natural area would be somewhere like a forest or any area with relatively low human disturbance. Next slide, please.
So this is an acronym for the most impactful drivers that determine soil differences between our areas. And this was our basis for experimentation and a major part of our research. So the CL stands for climate, the O stands for organism, the R for relief, the P for parent material, and the T for time. Next slide.
One factor that we realized affects soil but isn't part of the CLORPT acronym is human impact. Not all of the world's soil is undisturbed in a peaceful forest or desert. Much of it is in lawns, under buildings, or routinely receives a dose of fertilizer or pesticide. As a research group, we are really interested in what effects humans have on soil and why this is significant for the environment. Next slide. Much of our daily life requires soil to be altered. The food we eat was grown on a farm or raised on grazing grounds. We obtain materials from mines or deforestation. Even our lawns and gardens are altered from their natural state. Each member of our group sampled soil from a natural environment and from a disturbed environment, which for us was usually our lawns or our gardens. We are interested in how the soils compare in their organic horizons, biodiversity, and root density. Would anyone from the audience like to share a prediction on what we found the differences between disturbed soil and undisturbed soil for any of these three factors?
Any audience members can feel free to unmute themselves and share.
Lower mycorrhizae in the disturbed soils.
Thank you. We'll see. Next slide, please.
So in order to compare the human impacted areas and the less impacted areas, one of our main methods of characterizing the differences between the soils was measuring the depth of the organic horizon. So the O horizon is the top layer of the soil, which usually contains organic matter like decomposing leaves. And so in each of our plots, we dug until we reached the mineral horizon, which often has a different color and composition when compared to the organic horizon. And then we measured the depth of each of our holes. So these box plots show the centimeters to the mineral horizon in the plots with more human impact, which you can see on the left, and the plots with less human impact, which is on the right. In 8 out of 10 of the sites that we sampled, the organic horizon was shallower at the plot with more human impact, and the average depth for the human impacted areas was 10.8 centimeters compared to 21.5 centimeters for the unimpacted areas. And there's also a smaller range across the sites for the human impacted plots. Next slide, please.
So what I measured was the changes in biodiversity in the areas of human impact and the areas without human impact. So this is the percent increase in the plants. So around the different parts of the country, you can see how in certain areas there's a 0% increase in the biodiversity of plants, and some there is 600% increase. But what we measured with the plants was the plants that were in a foot by foot square or meter by meter square, and the roots that could be going underneath of it was counted in the area. Next slide, please. Did I say next slide? I'm sorry, I can't remember if I said it or not. All right, so the next was the biodiversity of animals. So these animals were, we didn't want to limit ourselves to just insects because there were worms and other things like that. So what some of us did is we laid out pitfall traps. So that's a cup or a dish of water with a little bit of soap in it that's buried on the same level as the soil, so that any bugs will fall in and the soap will break the surface tension and then they'll fall in. So when I left mine overnight and I came back and I counted different numbers and different species of insects and other animals that had fallen in. So here are the percentages of those areas from the altered to the unaltered. Next slide, please. And then here is the increase in the biodiversity of the types of worms. And why worms are separate from the animals total is worms are a huge factor for soil aeration. Worms form tunnels in the soil and they transport nutrients and they are very important for soil health. Next slide.
So what I did was go and research the various parent materials that make up our respective areas and finding out what they were originally made out of, or what the lower areas in the soil are made out of, as well as any glacial history, which also does affect the health of the soil. And along with affecting the biodiversity with the type of soil that it is, you can also see that there's a very large change between which side of the country you're on, and there's a lot of very large words here. So basically, where I'm just going to use the little one at the very bottom left, because that's the one I can explain the best, is my current area. Weakly consolidated alluvial sediment, so the ground is very hard packed, it is very rocky, it's very gritty, has a lot of loam and silt in it, and that just makes it very hard and rocky along with the arid climate. So areas that are glacially affected tend to be either more fertile or have a greater rate of biodiversity due to the effect that a glacier causes in a landscape. Then there's also a whole different type along with the coastal landscapes too. So yeah. Next slide.
We measured the root density of the soils by looking at the ratio between the volume of the roots that we dug up and the volume of total landmass removed. The higher the soil density, the more roots the higher the root density, the more roots there are in the soil. As you can see in the graph, the green bar represents the undisturbed soil and the yellow bar represents the human impacted soil. We found a much higher density of roots in the undisturbed soil than in the impacted soil. Since organic material is made out of carbon, having roots in the ground means that more carbon is sequestered in the ground and there is less in the atmosphere causing climate change. Also, roots often pair with mycorrhizal fungi, so more roots in the ground allows for decomposition in nature's recycling. When humans impact soil, they subtract from these essential natural processes. Next slide, please. Taking a look at all of our findings associated with human impact, it is clear that keeping soils undisturbed is important. But take a look at the graph. Our population is growing at a very high rate, and each person has a carbon footprint. As more and more of our land is being disturbed, it is critical for us to learn about the consequences that that may have. Next slide.
Hello, so this is Quinn, and I worked partly on analyzing how do climate factors affect soil. So next slide. And this is the data that I compiled from all of our documents. And the MAP stands for meters of annual precipitation, while MAT stands for meters of annual temperature. And we took these data from NOAA, which is a US database, and we compared it to the depth to mineral soil at our undisturbed and disturbed sites. So the red points represents less human impact and blue represents more human impact. And for looking at the precipitations in relation to depth to mineral on the left, we could see that there's a positive correlation. And while on the right with temperature, there's not as strong of a correlation. And this might be, so this shows us that precipitation plays a critical role in how depth to mineral is affected. And why this is important is that, and how it relates to JSEP and climate change is that as our precipitation, our precipitation and temperature can change as the temperature fluctuates due to climate change, and that could impact the depth to mineral soil, which is really critical to the way plants prosper. And next, please.
One of the major climate factors we thought may have an impact on soil biodiversity was precipitation. So we collected data from the National Oceanic and Atmospheric Administration on the mean average precipitation in centimeters of our different locations, and we graphed it with the biodiversity of our areas to see if there was a correlation. And so we quantified the biodiversity by counting the number of species in our soil samples that we collected. So based on our data, there seemed to be no correlation, but this might not reflect the relationship between biodiversity and precipitation in reality. So for more accurate conclusions, we would probably need a larger data set. And I also think it's pretty interesting that we got different results on a similar topic compared to the fish team.
So as a continuation of the climate and how it affects the soil, I made some graphs about the climate and how it affects the root density. Really sorry about the quality of them. They're a picture on my phone of a computer screen due to some technical difficulties. So you can kind of see there is not a very good correlation here. Oh yeah, and the blue is the more human impact, and the red is the less human impact. Between the mean annual precipitation and total root density. I think I've. Oh no. Wait a minute. Oh, okay. I am talking about this. Okay. Yeah. So some things that I found that might appear on the layer is that perhaps as there is more, you can see there's like kind of like a slight decrease. I'm not sure if that's an actual thing or if that's just something that we found. It might not be a connection. It's just a maybe correlation. Is that maybe as there's more rain, the roots might be smaller because they don't need to be as big to soak up the rain, which is something I guessed. But it could also be we have limited data, and I don't know. But the American Geophysical Union also said that for water-limited ecosystems, increases in wetness of the climate produced deeper roots, and the root depth is sensitive to changes in the depth of rain events than to their frequency. So yeah, next slide. This is the climate and root density, which is measured in temperature Celsius. I noticed that, yeah, not the best the data isn't really solid, but it looks like there's a little more of a trend here. As the temperature goes up, there's more root density. Yeah. Another thing to consider in this that we didn't consider is that we took different amounts of soil samples and like separated the roots from them, and then like did like a rate of like, did like a percentage and found the percent of root. And we didn't consider, like, some people took a lot more soil than others, like what it was coming from, like the organic horizon. Maybe some of it people got like from also the mineral horizon, because some people dug deeper. And we also had, like Sam was saying earlier, a lot of different types of soil, which are also factors. Yeah. Next slide. Thank you.
Hi, my name is Anna, and I focus more on the polar science discipline of climate and soil, and so this is another subtopic that we went on in this module. Next slide, please. So in our data spreadsheet, we all also researched independently our glacial history and if there was any. So basically asking, was there once ice that covered our plains? And so here are the 4 states that have the most prominent glacial history, and we just kind of detailed when the last movement was. So that's what all of the numbers means, like 12,000 years ago, 20,000 years ago was the last prominent movement of glaciers and ice. And so this carved out a lot of our regions and deposited some sediments and residue remains and accumulations of silt. And so once we started to compare this, we can now look in the future why all of our regions have different soils and what this means for our climate change today. Next slide, please. Awesome. So now that we've looked at the history of our past of these 4 states, I was able to take out the 4 most common remains and residues that were left behind. And so these were moraine, glacial till, glacial outwash, and eskers. And what these 4 are moraine is glacially formed accumulation of unconsolidated glacial debris that occurs in both currently and formerly glaciated regions on Earth through geomorphological processes. And this basically means like a ton of sediments are all compacted on our coasts, or if your state has more hills or regions, then there's a good chance that it was carved out by glaciers that were moving a couple thousand years ago. Glacial till is a subregion of moraine, which is like the small little peat and clay-sized fragments and boulders, and it's just a jumble of rocks and sediments that was mixed into the ice. Which is more of a heterogeneous mixture compared to glacial outwash, which is more of a homogeneous mixture, and that's sediments and stones that was freed by melting ice and the sands and silts. And then last, we have another formation called eskers, which are also deposited along the course of ice rivers that were moving as they were melted. So this is why when you're asking why are all of the regions of the US so different, you can take a look at their glacial history, and as soon as you start taking a deeper look into that, you can realize why there are all these different depositions and accumulations is because of what glaciers moved or didn't move and deposited these big rocks. Next slide, please. So now that we know our glacial history, you can sort of tie this into what the other students were talking about with soil quality. And now that we understand our impact in the history, we can break down why we need to maintain our soil quality. And I know that we're running low on time, so I'll try and be as succinct as possible. While weather is a short-term part of climate, certain weather cycles can still affect soil. For example, soil can be dried out and rearranged during droughty or windy weather. As the soil is dried out, plant growth is reduced, which reduces the stability of the surface layer and allows more erosion. An extreme example of this is called desertification, and that's when soils are losing their moisture and become more silty and sandy, and this is what leads to things like the Dust Bowl, which was during the Great Depression. So to avoid these phenomena, that's why we started looking up how to retain our soil quality, and the top ways that we found that we can do this are reducing the number of animals grazing, moving animals to newer areas more frequently, using conservation tillage and no-till practices, using low intensity and high diversity crop rotations, maintaining perennial vegetation, using irrigation properly to increase ground cover, and using selective or staggered tree harvest techniques rather than clear cutting. So overall, soil is a crucial part of the carbon cycle, and it changes in soil management practices can reduce emissions of carbon containing gases from soil, which reduces climate change and reverses global warming. Thank you. Next slide, please. And these are just some pictures of us going out and researching. So that's pretty much it. Thank you so much. Next.
Great, thank you so much, soil team. If we can do an unmute and give a round of applause.
Ooh,
Yay.
go soil team.
And as Erica is transitioning slides, if there's a quick question, you can put it in the chat, and maybe we can get one question in before we start the last presentation. And for all of the students, all of these great photos and drawings, there's a request from the JSEP PR team to make sure you share those so that way we can share what we've been doing on social media. So I think for the sake of time, we'll get going with the final presentation today. This is a presentation, the Joint Sounds Enable Progress presentation made by a group of students based in the US and also students based in Greenland. And I believe the format of this will be a little different. The students will share what they did, and then they're going to direct you to a product they created to explore for a few minutes online, and then we'll come back for a wrap up. So go ahead and turn it over to the sound group.
Hi everyone, my name is Helena and I'm from Cleveland, Ohio. And so we took a bit of a different approach, as Lauren explained. Instead of collecting traditional field data, we did a more abstract project and where we incorporated a bit of the arts and recorded and analyzed sounds in our environment. So we are the Sound Research Group, or the Joint Sounds Enable Progress Group. So earlier in the week, as part of one of our exit tickets that we complete each evening, we were asked if we speak any languages other than English. Some people spoke 2 languages, some people spoke 3 languages, some people even spoke 7 languages. but all of you answered wrong. You all speak one more language, and to all of our guests, you speak one more language too, and that's the language of sound. Next slide. So we have this quote by Hildegard Westerkamp, and she's a Arctic scientist. And it's, "I hear the soundscape as a language with which places and societies express themselves. In the face of rampant noise pollution, I want to be understanding and caring of this language and how it is spoken." Next slide.
The first step into hearing what is spoken is to be quiet and let it speak for itself. To make it heard, we decided to make a sound map, which is a representation of different environments and locations. With sound maps, you can travel the world just by listening to our recordings.
We went to record specific we went to record specific places that are close to us here in Kangerlussuaq. We drove and hiked to different places such as the ice shelf, Russell Glacier, to the rivers, and to the airport, which are the loudest man-made sounds here in Kangerlussuaq. The reason we decided on those locations is because they are the most exciting places where we are. They are also unique because there isn't a lot of places you can hear these specific sounds being spoken. Some also edited their recordings to remove themselves from the sounds, such as the sniffles and mumblings. Next slide, please.
Hi, my name is Armilla, and I'm going to talk about more about our sound map. You can go to the next slide, by the way. We use spectrograms to help visualize our sound data. And basically what a spectrogram is it uses different colors that corresponds to certain frequencies of sounds. So we get this visual representation of what's going on in the sound that we recorded.
Hi, I'm Elise, and I'll also be talking about this. So the picture on the bottom left is one of the spectrogram recordings that we took. The pink lines represent distinct sounds and higher frequencies of sound. So for example, these lines that you can see are a bird call. You can see the sort of musical pattern of the noises that they make. And the picture on the right is another representation of a spectrogram, but this one was created with higher quality equipment and microphones that we didn't have access to. But you can also see again the pink frequencies representing the sound around us. Next slide, please. So why do we use spectrograms? Since we are visual processors, it helps to present our data in a visual way. Similarly, we aid our studies and data with graphs and charts, and it helps us analyze the various calls of animals around us, and it highlights patterns and outliers.
So the two spectrograms at the bottom were both taken at the same location, but one was taken in the morning and the other in the afternoon. And you can see in the morning there are more high-pitched frequency sounds than in the afternoon, and those are because in the morning there are more bird calls. So it goes to show that these spectrograms are really helpful in aiding seeing the visual patterns over a course of time. Next slide.
So there are these things called sound walks, and there are two different kinds of sound walks. There's a narrative sound walk and a non-narrative sound walk. And in general, a sound walk is a guided walk that focuses on listening to a specific environment. And in a narrative sound walk, the listener is guided by listening to someone else's voice, and they explain the sounds being heard and maybe the person's personal connection to the sound. And a non-narrative sound walk is the same thing, just without words, and you can hear what the person or where the person is walking, like the leaves crunching under their feet or something like that. And it's just another way to connect to the environment. Next
Elise, do you want to jump in?
Yeah, I
Elise
can.
is our understudy, everyone, just in case.
So there are 3 different types of sound. So first we have keynote sounds, which are sounds that are created by a landscape and its elements. And then we also have signal sounds, which are often man-made sound events that we listen to actively. So cars going down the street, airplanes above you, stuff like that. And then we have sound mark sounds, which are a sound that is specifically noted or regarded by that community. So in each of our communities, something that we're really used to, like a specific bird call that we always hear in the morning, etc. And we recorded some sound mark sounds that are going to be a part of our sound map. Next slide.
This topic is intended to demonstrate the effect humans have on our environment through noise pollution by contrasting environments with noise pollution to environments with little to no noise pollution. We combined recordings of natural sounds with recordings of unnatural sounds.
Hi, my name is Daniel, and I'll also be presenting this slide. I'm from Florida. And just as Ms. Westerkamp said at the beginning of the presentation, in her quote, noise pollution is a huge issue. Humans are accustomed to the sounds of nature, and not hearing those sounds is kind of jarring for us. Therefore, we were able to distinguish a human sound from a natural sound, and this is an observation we wanted to reflect in our sound map. And as seen by the pictures, we even try to imitate the natural sounds, but it's not as kind to the ears. Next slide, please.
So hi, I'm Charlotte, and I'm going to be talking about another aspect of our sound map, which is our recordings that show time passing. So the sound map we created includes them over a period of a day, but often they are used to show how sounds change in the same location over months or even years. So it's a tool that can be used to determine what factors are affecting the natural environment, like noise pollution from human interaction, and also to learn about natural patterns through different seasons and times of day. So some animals are active only at night, or sounds can be different with the presence of crunching leaves in October or snow in January. So it is a tool for analyzing these changes and the amount of change in one climate or environment compared to another. Next slide, please.
Hi, I'm Aya, and I'm from New Hampshire, and I'm going to be talking about the overall purpose. So our JSEP map acts as a multi-purpose application focusing on entertainment, education, and research. So why does the JSEP sound map matter? Travel can be expensive, and the emissions hurt the environment. The JSEP sound map empowers people by giving them the opportunity to transport themselves around the country and even internationally to Greenland without having to pack their bags, pay money, or harm their environment. We are also fortunate enough to have such a wide range of locations within our team to help display all the different unique environments and cultural sounds. In addition, this project has multiple different functions which have been shown throughout this presentation. So how can sound prove and provide new information about climate change? Our hope is that future scientists can use our soundscape to listen to the past and notice the differences over the years. Climate change can be proven through the fact that the world's soundscapes are changing for the worse. Diversity and natural noise are slowly fading and being replaced by artificial mechanical noise pollution. So last but not least, with everything that's going on right now, coronavirus does not have to stop travel. Please use this for your enjoyment and take a look at our map. Some notes are: don't forget to use a computer for easy and efficient access, and zoom in on specific locations because there will be multiple audios in what might sound like one spot. Thank you so much for listening.
So in the chat, Clara Allison, the mentor for this group, has posted a link. So we want everyone to take about the next 4 to 5 minutes to go visit this sound map, listen to this product that the students have created, and then I'll call everyone back in about 4 or 5 minutes. And I think the group has a couple of concluding thoughts, and then there will be time for questions. So for now, just go to the sound map. You may want to mute your microphone and check out this really cool website. Okay, I hate to disrupt all of the listening going on, but I would like to ask everyone to rejoin for a wrap up of this presentation, and you can continue exploring the sound map at the end of the presentations. So I'll turn it back over to the sound group for a few closing remarks.
Thank you. Our map will stay up online, and we hope that you guys spend more time looking at it. We just wanted to say, although JSEP was challenging this year because we all had to be separated and we couldn't be together in Greenland, we utilized the different locations to our advantage, and we're so grateful for this opportunity. Thank you so much for listening, and we're open to any questions.
And Natalie, if you could unmute us for a final round of applause for the sound group and also just for everyone.
Yay!
Ooh.
Very well done. And I want to leave a little bit of time too for any questions for the sound group. There's one question for the sound group, if they have any data on how sound affects mental health and environmental health.
I mean, it's not necessarily like environmental sound, but for the mental health, like part of the question, I know when like usually more environmental, like natural sounds are like calming, and it's like proved to help you be more mindful and stay in the moment rather than a lot of mechanic sounds and a lot of like human made sounds that can be very distracting and overwhelming, and I know can like are actually proved to cause anxiety. So I think that's just another like reason why it's important to like connect with nature, and I think that's what all of these presentations have kind of been about.
Yeah, in addition, for environmental health, like noise pollution is definitely a huge problem. Not only does it like distance us from nature in many ways, it can also like affect any animals living in the area. So yeah, like trying to prioritize like having less noise and like saving time for more quiet natural sounds is definitely a good thing.
I know do you think you captured any changes due to COVID? I think because there have been less humans outside, we definitely have been hearing more wildlife, especially in my own backyard. The animals are coming out more and we can hear them. And then also there were like places like parks that I wanted to visit and record sounds, and unfortunately they were closed due to coronavirus. So it's been affecting us, but we made it work.
Thank you, Aya. There's one last question. Do you plan to add more sounds to the map throughout the year?
That's such a good idea, definitely. We have to figure out how to add them, like, since we aren't going to be like in a program, but yeah.
Yeah, we hadn't talked about it, but honestly, that'd be super sick. And if we can get, we sent out a call to like all of the JSEP participants asking for people to send in their sounds. And I know a lot of people were busy like doing their own thing, so they didn't get a chance to, but it'd be cool if we could get even more from like across the country.
Yeah, we can definitely work on making this open source if people are interested.
And it would also be cool to add more sounds because it'll be over a greater span of time, because what we have here was only like 2 days. So if we had more sounds over, like say if we continue this into like next year, with JSEP next year, it could actually like indicate some type of environmental change perhaps.
And with our Danish students, we'll get even more diversity if they can send us some recordings from Denmark, because I know some of them live in Copenhagen or really small towns, and that's a place that like not many of us have heard, which I think is really interesting.
And while we have such an amazing network of JSEP alumni here too, again, it's so wonderful to see so many familiar names. If you're interested in contributing to the project, get in touch, because it would be really fun to build this out and make it an ongoing JSEP project. So we are out of time. I just want to say these presentations were fabulous and very inspiring. And I hope before you all leave this room, you spend a few minutes scrolling through the chat. There's some really great feedback, more questions, lots of interaction. I think that I'm getting the sense the audience is very interested in everything you've had to say. So Natalie, if you can unmute us for one more round of applause.
Yay.
Ooh, 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 ooh. Amazing. Amazing.
Very well done. And so this concludes our JSEP 2020 presentations. And so I'll be in this room for just a few more minutes, and then we'll unfortunately be closing this out. But for all of the students in this group, thank you so much. Job well done. We have our JSEP wrap up immediately after this. So all of the students, we ask to make sure you rejoin this room in about 15 minutes. And thanks again to all of our guests for being here and hearing all of this really great science and art. So thanks for being here, and see everybody soon.